So, good afternoon. My name is Konstantina Trivisa, and I'm the director of the Institute for Physical Science and Technology at the University of Maryland. It is my pleasure to host the 28th Shih-I Pai lecture and to welcome all of you to join us. So today we honor Dr. Shih-I Pai's many accomplishments and contributions to science. A pioneer in the study of turbulent flow and its effects on the drag and lift of aeroplanes, Dr. Pai served on the faculty from 1949 until he retired with emeritus status in 1983. A founding member of the Institute for Fluid Dynamics and Applied Mathematics, IFDAM, now known as the Institute for Physical Science and Technology, he published 14 books and 130 articles in the fields of aerodynamics, fluid dynamics, and viscous flow, for which he received international recognition. Thank you, Pai family, for carrying on his legacy. It is my great pleasure to introduce our speaker today, Cristina Marchetti, who I met some time ago in Germany, so it is a distinct honor for me to introduce her today. Cristina Marchetti is a theoretical physicist who has worked on a broad range of problems in non-equilibrium statistical and condensed matter physics, including supercooled fluids, gases, superconductors, and more recently, her research focuses on collective behavior in condensed matter and biological systems. She has played a leading role in the development of the field of active matter, which is the topic of her lecture today. After postdoctoral appointments at the University of Maryland, Rockefeller University, and City College of CUNY, she joined the faculty at Syracuse University in 1987. In 2018, she joined the physics faculty at the University of California, Santa Barbara, where she is a faculty today. She was awarded a number of honors, a Rothschild Mayen Fellowship at the Institute Curie, a Simons Fellowship in Theoretical Physics, and the inaugural Leo Gadenoff Prize by the American Physical Society in 2019. She's a fellow of the American Physical Society and a member of both the American Academy of Arts and Science and the National Academy of Science. The title of her lecture today is The Physics of Active Matter. Cristina, the floor is yours.
Thank you. Thank you, Konstantina. Let me just take a minute to share my screen.
Yes.
Now it's not working. Okay. Sorry about that. What happened? Okay. I'm sorry for a second, there's a technical issue here. I will solve it.
You are in presenter mode, Cristina. There you go.
Now,
Okay. Well, thank you, Konstantina, for the very kind introduction. It's a true honor for me to be here today and to be asked to give this lecture to celebrate Dr. Shih-I Pai. And it's also a true special personal pleasure because the University of Maryland, and specifically IPS, the Institute for Physical Science and Technology, is where I learned my non-equilibrium statistical physics. I was a postdoc there back in the 1980s. And as a postdoc, I really had the fortune to learn about this wonderful field from two masters of the field, Bob Zwanzig and Bob Dorfman. They were my postdoctoral supervisors. And I also had the pleasure to work with Ted Kirkpatrick, that at that time was just starting as a junior faculty member there at the university. And in fact, to this day, I ask all my graduate students to actually read this little book by Bob Swansik, which in my opinion is one of the best and most concise presentations of the topic. And so it's really with great pleasure that I get to give this lecture to celebrate Professor Pai and tell you about the field of active matter, a field that brings together ideas from fluid dynamics, non-equilibrium statistical physics, pattern formation, dynamical systems, and more. And so it seems really particularly well suited both to celebrate Dr. Pai, as well as to celebrate itself, which really is a place that has pioneered research that crosses disciplinary boundaries. So with that, I would like to start with a little bit of history. Physics is able to describe how atoms and molecules aggregate to form a variety of materials, fluids, solids, with properties very different from that of the individual constituents. And so physicists some time ago asked the question of whether non-equilibrium statistical physics can actually be used to describe the collective behaviors such as bird flocking, where many birds, like in a starry murmuration, aggregate and fly together in a coherent manner and move in the sky as a soft fluid. And really wouldn't be right to give a lecture on active matter without showing an example of such bird flocking. So let's just watch it. So back in 1995, Hungarian physicist Tamáš embarked in this quest of trying to describe this kind of behavior using physics ideas. And he proposed a simple model that indeed captured the flocking of birds. Now, truth be said, the model was already in the literature because Craig Reynolds, a computer scientist working for the animation industry, had actually introduced this model quite some years earlier and had used it in a number of feature movies such as Batman Return, and in fact had won Oscars, as won an Oscar for his work. But what distinguished the work of Vicheko was that he really was able to show that flocking could be understood as what in physics we call a phase transition. And the model that he introduced is based on an analogy with ferromagnetism. In other words, he modeled the bird as a velocity vector or an arrow, if you like, a flying spin, in analogy to the spin carried by magnetic atoms. So the velocity, the speed, the magnitude of the velocity is fixed, and the only degree of freedom is the direction in which the bird flies. So these are really point particles, and the model says that each bird aligns with its neighbor within a circle of prescribed radius with noisy rules. That is, it makes some mistakes in this alignment. And these are the equations of the model. So the angle in 2 dimensions, the direction is just an angle. The angle is updated by aligning with the neighbors with some noise, and then the bird moves forward with a fixed speed in the new direction. What we call the order parameter, which quantifies the transition, is the mean velocity of all the birds. And what one can show is that these models exhibits a transition from a state either at low density or high noise, where the mean velocity is 0, or the birds fly in random direction, to a state at high density or low noise, where the mean velocity is finite, in fact, of the order of V0, and all the birds fly coherently in the same direction. And this state is really like a polar flocking fluid. So this is a numerical model. You simulate it on a computer, and you see the flocking of the birds. And the color here denotes the direction in which the various groups of birds are flocking. Now, just in the same year, actually, John Toner and Shih-I Pai, working together, also developed a continuum model, a hydrodynamic model of the same phenomenon. In some sense, the fluid dynamic model of flocking, by writing equations, continuum equations, that couple the density of birds to their mean velocity. And the way they formulated the model is essentially by merging a well-known equation of fluid dynamics, the Navier-Stokes equation, with the dynamics of magnets. And a key feature of these equations is that the velocity, which is the order parameter of the model, advects the density. And as a result, this system has very large density fluctuations, and that turns the phase transition into a first order phase transition, very much like the transition between liquid and gases. Now, so what you have here is a situation where the motility can stabilize through long range order in 2 dimensions, which is unusual because in equilibrium, we know, physicists know, that you cannot have true long range order in 2 dimensions. This is a specific feature of non-equilibrium systems. Now, so the lesson of Vichek's model, if you like, is that you can actually obtain organized behavior of large noisy collections of self-driven entities without a leader, and it can emerge from simple local rules as opposed to, say, long-range interaction or biochemical signaling. And this idea, this framework that he proposed, has had since then has been applied on many, many different scales, from describing the structures inside living cells to the behavior of even human crowds. So with that, let me actually now go and tell you a little bit more generally what we mean by active matter today. So today, an active particle is essentially an engine that consumes energy to generate its own motion and forces. Example is a bacterium that uses nutrient to swim through a fluid. There are actually synthetic realizations, colloidal particles that are propelled by chemical reaction, and I'll show you an example a little later. And so active matter is a collection of such interacting energies and engines, which as a result of interaction organize in new states, which you can really think of as new states of matter. And as we will see, there'll be flowing fluid or new structures with unusual properties. And this kind of behavior can be found in both the living and the non-living world on many, many different scales. Starting from structures inside living cells. This is something called the mitotic spindle. It's a very special structure that actually drives the separation of chromosomes when cell divides. There are engineered microswimmers that we call active colloids, and I'll show you an example of those later. Of course, bacteria, living cells, collection of small robots, and then even at our scale, birds, fish, and as I said, human crowd. So what is common to all this system is that there is an energy input at the scale of the individual particles, and this energy cascades to large-scale dynamics and organization. An example is, for instance, protein like myosin, which are a class of what we call motor protein and sit inside your muscles, use chemical energy to eventually transform it into forces that control how we move and function at a much larger scale. And the defining property of these active systems is that, again, the energy injection is at the level of each individual particle and independently on each individual particle. So, but to understand a little better how is active matter different from systems that are driven out of equilibrium in more conventional ways, for instance, a sedimenting suspension or a fluid that is subject to shear at its boundary and driven to flow by applied shear. let's actually go back to something that is probably familiar to everybody, which is something called Brownian motion. The fact that if you have the famous grain of pollens in water or in air, that if you look at them under a microscope, you see that they are continuously undergoing this jiggling type motion, moving around as if they were alive. And if you were to draw the path of one of these particles, it might look like something like this picture here. I hope you can actually see my cursor. I think you should be able to. So a Brownian particle experiences both a mean drag due to the interaction with the surrounding fluid, and the surrounding fluid also yields driving forces that come from the constant collision with the water molecules. And the driving forces are proportional to the drag, which I called gamma here, and to the temperature. So if the solvent fluid is in equilibrium, the solvent fluid yields both the drag and the drive. And these two are related. And that's the statement of something that we call the fluctuation dissipation theorem, or we say that this system exhibits detailed balance. And that leads a long time to the fact that the motion of these particles is grows linearly, the mean square displacement grows linearly with time, the motion is diffusive, and the diffusion coefficient is related to the drag, to what is known as the Einstein relation. Now, an active particle, it's a bit different. It also experiences a drag from a mean drag from the fluid. It also experiences driving forces from collision from the water molecules. But in addition, it has an internally generated drive that actually far exceeds in strength the drive due to collision with water molecule, which are therefore essentially negligible in this case. But because it has this internal driving force that actually drives the bacteria, in this case, to swim, in this case, the drag and the drive are not balanced. They don't come from the same source. And so there's no fluctuation theorem, no detailed balance. And the dynamics of each one of these microswimmers is not time reversal invariant. So this drive breaks time reversal symmetry locally and independently on each particle, and not globally, like, say, an external electric field would if these were charged particles. And that's really what makes the system very different. We now have a collection of interacting particles where time reversal symmetry is broken at the micro scale. And therefore, this active system can generate their own currents and their own self-sustained flows. So what we have when we have a collection of these active particles is the new kind of non-equilibrium matter made not of atoms and molecules, but of entities that generate their own forces and motion. And then we can ask a number of questions, such as what new states are possible? What are the quantities actually we tune to change from one state to another? In equilibrium, we know that we can tune density and temperature. We tune temperature to go from water to ice, say. But in this type of system, we will see there are new quantities we can tune to change the behavior of the system. For instance, the motility of our swimmers. Can we classify the behaviors and identify generic properties? And are there new classes of active systems, such as we have in equilibrium? Now, because we are out of equilibrium, we cannot start with a free energy, but we have to go directly to the dynamics. And furthermore, there is an important theoretical challenge that this system provides, which is that really we don't have a general rule for formulating the statistical physics when there is no detailed balance or time reversal symmetry at the micro scale. And so we kind of have, at the beginning at least, we had to kind of start it with the system sort of one by one. So what I want to do in the rest of the talk is to give you two examples of the new kind of physics that arises from motility. The first one is has to do with the fact that the system can exhibit spontaneous assembly without any attractive interaction. Without attractive interactions, and I'll do that using a very minimal model of active matter. Physicists are very fond of minimal models that, although minimal, actually can apply to colloids and bacteria. And the second example will be one about fluids that flow spontaneously without externally applied forces or pressure gradients, and that spontaneously exhibit what we call active turbulence. And I felt that this was a particularly appropriate example, given that Dr. Pai himself made important contribution to the field of turbulence, although, as we will see, a different type of turbulence. So let us start with the first example. A minimal model we can make of an active particle is what we call an active Brownian particle. These systems, such as bacteria, are generally large, micron size, and inertia is generally negligible, so we neglect inertia. So the equation of motion, instead of being Newton's law, F equal ma, will just be essentially net force equal to 0. So we model the active particle as a spherical bead with the self-propulsive force of fixed magnitude along a body axis, which I denote by n hat, that can change its direction due to random noise at a rate 1/tau. So if you want to look at the equation, they're very simple. The velocity is simply what we call the self-propulsion velocity, fixed speed and direction randomized. Direction, which is here just an angle, because here is just 2 dimension. The angle randomized by noise at a rate tau, where this eta is just a white noise of variance 1. So this particle will sort of travel in a straight line ballistically with a speed v0 for a time tau, and then change this direction. Now, there are actually realization of this type of system. This is what I'm going to show you in a video in a minute, is a micron size, basically plastic bead, which has embedded in it a piece of a magnetic material called hematite. Hematite, and it's immersed in a solution containing hydrogen peroxide. Hematite catalyzes the breakdown of hydrogen peroxide into hot oxygen and water, but it only does so when you shine UV light on it. So in this video, what you are going to see, the video will tell you when the light is on and the light is off. What you will see is that when the light is off, these particles behave like Brownian particles. They just jiggle around. But when the light is on, they actually become swimmers. They swim around like bacteria. In fact, at speeds pretty much comparable to the speed at which, say, a bacterium like E. coli swims. As you see, the light is off, they just jiggle. Now the light is on, and they actually swim, just like swimmers. This was work done some years ago in the lab of Paul Chekin at NYU. So my minimal model of an active Brownian particle is actually a fairly realistic one for this type of microswimmers. Now, a long time, the dynamics of one of these microswimmers actually is diffusive, with a diffusion coefficient, which is given by the square of the speed times this time, which we call the persistence time, because it's the time over which these particles travel ballistically in a straight line. And so then it would be tempting to say, well, then we can use Einstein relation and define an effective temperature for these particles, which turns out to be much larger than the physical temperature, but it's very tempting then to say that these particles seem to be just hot colloids. They behave like just hot colloids. What is so special about them? Well, what is special can be seen when you put many of them together and you add interactions. In this case, just short-range repulsive interactions. So these are just like billiard balls with this funny, persistent, random walk type dynamics. They go in a straight line over a persistence length, and then they're randomized by noise. But now they also have repulsive interaction, and their activity, as we call it, can be quantified by a dimensionless number, the Peclet number, which is essentially the ratio of the persistence length to the particle size. If you simulate this model on a computer, what you find is that these particles with purely repulsive interaction actually condensate. In equilibrium, you require attractive interaction for a gas to condensate into a liquid. Here, there is just repulsion. And as you can see, these particles condensate into this big cluster surrounded by a gas of active particles. This phenomenon, known as motility-induced phase separation, is actually a very generic phenomenon, has been verified by large-scale simulation, and we have a very well-developed continuum theory that has been obtained by coarse-graining the microdynamics of this particle, as actually I used to do when I was working at IPST with Bob Dorfman. And we can actually understand it rather simply in at least a couple of ways. The first one, from sort of a kinetic theory point of view, is that what we're really seeing here is a traffic jam that occurs when the persistence time is large compared to the typical collision time between, or mean free time for collision between particles. In other words, what you find is that when a particle joins a cluster, and there's no time, the persistence time is long, so it has no time to turn around and leave. And as a result, the flux in is much larger than the flux out. And in other words, the system breaks the tail balance, and there is really a traffic jam. These particles start aggregating because they have no time to turn around and leave the cluster. Another way of looking at this is that there's actually been, as I said, a detailed theory constructed. You can actually show the crowding suppresses the motility of the particle, that is, the bare propulsive speed gets renormalized through interaction and becomes dependent on density and decreases at large density. And therefore, particles accumulate where they slow down. And in fact, what you can show is that there is a real spinodal instability. You can show that the steady state density for this particle goes like 1 over the speed that depends on the density. And particles tend to aggregate, which is really the opposite of what we are used to when we think about Brownian particle. Brownian particles like to diffuse and to go a situation of constant homogeneous density. And these active particles, instead, they aggregate where they slow down, and then there is a feedback that leads to this spontaneous aggregation. We can construct what in physics we call a phase diagram for these active particles. That is a plot in the plane of this dimensionless number, the Peclet number, and the density that separates the region where the system is a dilute gas, is a dense liquid, or it is in this phase-separated state where gas and liquid coexist, or at even higher density and not too high Peclet number, the system actually forms like a solid, glassy solid. And in other words, what we have here, this vertical axis, in equilibrium, the phase separation would have a temperature on this axis. What we have here instead is the motility essentially of the particle times the persistence time. And you can see that we cannot really think of motility as an effective temperature because actually things go the other way. The aggregation occurs at high motility in contrast to what happens in equilibrium. So what we have here, this motility-induced phase separation offers a new handle for tuning spontaneous assembly. And there's actually this minimal model I showed you has actually led to a lot of progress in understanding the behavior of motile particles, including the various phases, their behavior of the phases, and even constructing an effective thermodynamics of this system. Now, systems where the particles are not spherical or the interaction are more complicated are still somewhat of a challenge. Now, this motility-induced phase separation is a real phenomenon. In fact, going back to my plastic beads with the hematite, the self-propelled particles tuned by light, When you are at higher density, you can see that when the light is on and the particles are self-propelled, they indeed aggregate, just like in my minimal model of self-propelled beads. And as we'll see in a minute, that this is indeed due to motility, because when the light gets switched off, these clusters fall apart and disintegrate. And similar aggregation driven by motility is seen often in bacteria. So again, we have a new way of driving aggregation, which is simply motility and persistence of the single particle dynamics. The persistence is very important because it's what really distinguishes the self-propelled dynamics from the Brownian dynamics. Now, this idea that active particles accumulate where they slow down has actually been exploited in sort of remarkable experiments done by Roberto Di Leonardo at the University of Rome, where he used E. coli, so a strand of bacteria E. coli that is sensitive to light, and that becomes motile only in the presence of light. And by using lasers to generate patterns of light, he's able to get the bacteria to accumulate either in the regions where they're not motile, so in the dark regions, and to essentially paint with bacteria, as you will see in this video, where he actually switches from Einstein to Darwin and back by tuning the light pattern. The intensity that you see is proportional to the concentration of bacteria, and the bacteria are driven to the right place by using a light pattern that allows to change the concentration of bacteria by using this factor that they accumulate where they slow down. So this kind of work has led to a lot of progress towards engineering high efficiency micro and even nano machines with actually tunable and controllable motility. And let me now move on to my second example, that of spontaneous flow with no external driving forces. And I want to tell you a little bit about active turbulence. Now, I talked about spherical particles, but actually most living fluids are really not made of spherical particles. Bacteria generally, most bacteria are not spheres. They're kind of, at the very least, elongated objects. They're more or they're more complicated shapes. In fact, the shape can even change. So they're more similar to what in physics we know as liquid crystals. Liquid crystals are a fluid of rod-like molecules that, as you increase the density or lower the temperature, they can undergo a transition from a state where the molecules are randomly oriented, that's called an isotropic, and they form what's called an isotropic liquid, to a state where the molecules are on average aligned, oriented in a given direction, and that's called a nematic liquid crystal. These molecules have no head and tail, so in the plane of the page, their orientation is identified by a unit vector n that we call the director, but this is not a real vector. I know there are mathematicians in the audience. It's not, physicists can be a little bit sloppy with maths, so this is not a real vector. You can think of it as a two-headed vector because the up direction is the same as the down direction. So that's an important symmetry of this kind of liquid crystal. So this would be a passive liquid crystal, the material that is used, for instance, in your displays that you're looking at right now. An active liquid crystal is one that is made of elongated agents that are rod-like, but also exert forces on their environment. The example you're watching here is a dense suspension of a bacteria that lives in the soil called Myxococcus xanthus. And this is a bacterium that rather than swimming, actually these bacteria don't swim really, they glide on soil. And rather than gliding in the same direction all the time, they kind of go back and forth. and therefore they exert force dipoles, pair of equal and opposite forces in their environment. They don't have a preferred direction, but they have a preferred orientation. And so they form what we call an active nematic living crystal. Now, there are many realizations of active nematics of this type, and what is common to all of them is that they exhibit what we call active turbulence, these spatiotemporal chaotic type flows or turbulent-like flows, even though they are essentially at zero Reynolds number. Reynolds number is a well-known dimensionless number in fluid dynamics that measures the ratio of inertial forces to viscous forces. And typically, turbulence is observed at very large values of the Reynolds number, say 3,000 or more. Now, these systems all have essentially inertia is always negligible, so the Reynolds number is zero. And but they do exhibit a dynamics which is very similar to that of turbulent fluids and has been called active turbulence, even though this is really sort of a misnomer. So what I'm showing here is a variety of different systems. The first one is a suspension of filaments protein extracted from living cells, and I'll tell you a bit more about this system in a minute. This is a bacteria, E. coli. The third one are sperm cell, and the last one are actually human bronchial epithelial cells. And the reason for showing all these figures is to highlight the different scales and the different in both length, but also time scales. The cells are very slow. The numbers you are seeing here are minutes and seconds, and the typical phenomena that you're seeing really occur on the time scale of hours. So let's try to understand a little bit where this active turbulence comes from, and how can we quantify it and explain it. And I'll do that by focusing on this first example, tell you more a little about this first example over here. So these are active fluids assembled from protein extracted from living cell. If you look inside the cell, you see a network of filaments by a long protein that are filamentary protein. In particular, among those, there are ones called microtubules. This is a cartoon of a microtubule, and here is just a drawing of them. And these filaments are cross-linked by motor protein. Motor protein are, such as kinesin, are truly nano engines that are capable of transforming chemical energy into mechanical work. And they walk on these microtubules as shown in this animation. And for instance, they walk on microtubules and they carry vesicles filled with chemicals from one place of the cell to another. What one of my colleagues at UCSB has done a number of years ago is to engineer a fluid where he gets clusters of these motor protein to cross-link microtubules. And as the kinesin walks in opposite direction on the cross-linking microtubules, the microtubules will essentially slide relative to each other and extend out and exert forces on their surroundings. And you can see that in this video, the white streaks are bundles of microtubules cross-linked by this motor protein. And you see that they are continuously pushing out, extending, breaking up, and reconnecting. And so when you have a low density of this microtubule, you see all this sliding and reconnecting all the time. But at high density, what you see is this type of self-sustained flow that really looks like turbulence. And the turbulence is accompanied by the proliferation of this really strong distortion of so there's a lot of alignment here, but are these strong distortions, these things that almost look like comets, which are actually topological defects of the nematic structure. They're distortions that cannot be removed by continuous deformation. And there are actually two types, if you look closely, of these topological defects. There is the one that looks like a comet, and there's one that looks like a triangle over here. Now, in equilibrium, when these two defects these two defects would be attracted to each other and they would annihilate and form and disappear. But here they're continuously generated by activity, and in fact they drive this type of flows that actually mimic the kind of motion you see inside cell in the cytoplasm of cells. And if you actually plot out the vorticity of this flow, you actually see that there's really an important, a deep connection between the structure of the fluid, the structure of the nematic liquid crystal, the orientational order, and the properties of the flow. And that's a connection that we have exploited to try to understand this behavior. So where do this flow come from? Well, the flows are driven by what we call active stresses. As I said, as the kinesin walk on the microtubules, they displace these filaments relative to each other, exerting force dipoles. The forces here are always internally generated. There is no net force. They always come in equal and opposite pair, what is called the force dipole. And if you add up all the force dipole generated internally in this fluid, of course you get non-zero force, but you get a non-zero stress. So the net local force will be the divergence of a stress. And the stress, of course, depends on the filament orientation and is proportional to a parameter, alpha, that we call activity, that depends on the biomolecular processes that drive these flows. In this case, the concentration of the motor protein, and also the concentration of the chemical, the fuel called ATP, adenosine triphosphate, that is motor protein use in order to power their motion. Now, each one of these, so each one of these active dipole, these two equal and opposite forces that these microtubule exert on the surrounding, generate flow, and these flows are what drives the dynamics, the turbulent dynamics that you have seen in this system. Now, we can actually construct a fluid dynamics or hydrodynamics model of this behavior, and this hydrodynamics can actually describe quantitatively the dynamics in all these different systems, from the bacteria to the microtubule suspension to the living cells. The fluid dynamics is written in terms of a flow velocity, V, and essentially the director that describes the orientation of the nematic liquid crystal, or more precisely, the associated order parameter, which is really a tensor, Q. And the equation for the velocity is really just a Navier-Stokes equation, but augmented by elastic stresses associated with the fact that when the filaments are orientationally ordered, they actually have a stiffness, and so there is an elasticity to this system, even though it's a fluid. And most importantly, by these active stresses I showed you a minute ago, and this active stress acts as a source of flow and drives the spontaneously induced flow in the system. And the active stress itself is proportional to the filament orientation. And then we have an equation for the dynamics of the orientation, this director field, which is of course coupled to flow, because flow gradients and vorticity rotate and align the director, and then there is a stiffness for changes in the orientation of the director. These equations look complicated, but they can do many things. First of all, they predict that the order nematic is always unstable, is linearly unstable. This was actually something that was first predicted theoretically and then found experimentally. They predict that the order nematic is linearly unstable, and it's easy to understand why. Imagine you have an order nematic, it would just be a set of parallel lines, and then you have a fluctuation, what we call a band fluctuation. In equilibrium, the band fluctuation costs energy, and so the system will just go back to the order state. But in this active system, the constituents are these active entities that exert force dipoles on their surroundings. And these force dipoles generate flows. And so a band fluctuation will result in flows, as indicated by these red arrows, that actually tend to increase the size of the fluctuation, resulting eventually in an instability. And this is going to be shown here in this video, where the filaments have been aligned by flow. Flow has been used by alignment, and then the flow, and then let go. As you can see, they develop this band instability that gets larger and larger, and then eventually they start flowing around in this turbulent manner. And there is a characteristic length scale to this instability that comes out from this fluid dynamics equation I showed you a minute ago. Is essentially the length scale where elastic stress balances active stresses, and therefore is determined by the square root of the stiffness of the pneumatic liquid crystal to the quantity that I called activity. In addition, if you simulate, if you solve numerically this hydrodynamic equation on a computer, you actually reproduce these turbulent light dynamics that was seen in the experiments. What is shown here to the left is the structure of the director field, the structure of the nematic order parameter, with also the topological defects that I mentioned very briefly, shown as dots and triangles. And what is shown to the right is the vorticity of the flow, with blue and red corresponding to vortices flowing in opposite direction. And you can see a close correlation between the vertical flows and the distortion of the order and the director field. And in fact, the statistical physics of the system can be described either by focusing on the statistical physics of the defects or on the statistical physics of the vortices. And we have an understanding for how transition to turbulence occurs in this system. With increasing activity, if you had a system, is essentially everything is controlled by this active length that controls the linear instability. If you have a system of finite size L, when the active length is large, remember the active length goes like the inverse of activity. So a weak activity corresponds to a large active length. When the active length is larger than the system size, you will have an order nematic. When the active length becomes smaller than the system size, you can actually have laminar flow, spontaneous laminar flow with no applied forces of this active fluid in a channel. And this has actually been seen experimentally in layers of epithelial cells. And eventually, when this active length becomes much smaller, than the system size, you actually have this active turbulence that is really ubiquitous in all of this system. Now, one question we can ask in inertial turbulence, as it's called, has been studied extensively, and we have, in particular, there is an important result due to Kolmogorov about universal exponents describing the behavior. This is a plot of the essentially kinetic energy density in the active fluid. And in inertial turbulence, of course, the fluid is driven externally, and the length scale or energy scale at which the energy is injected through this external drive is very different from the length scale over which energy is dissipated through viscosity. So the energy is injected at large scale, is dissipated at small scale, and the intermediate range, we have this universal scaling with an exponent that is universal, which means it does not depend on the particular type of fluid or on the way in which you inject energy. Can we have similar kind of universality in this active turbulence? Well, that's actually a question that is still out to be completely answered, but there is certainly an important difference in active turbulence, which is that the energy is injected at the micro scale and then self-organized at the scale of this active length. So the pattern of energy injection is itself self-organized, and both the injection and the dissipation are really controlled by this active length. And so essentially the spectrum of energy in this case has sort of a single peak at the active length. In this case, there is a second length scale because this is actually a layer of fluid on a substrate. And so there is another dissipation controlled by interaction with the substrate, but this range is very, very small. And it's unclear at the moment whether any of this is the scaling of the kinetic energy for the particular microtubule suspension that I showed you earlier. And there seems to be certainly some universality in the exponents found at large wave numbers at small scales, but we're really not at the stage of being able to classify active turbulence in similar way as we have understanding for inertial turbulence. I'm almost done. I just want to leave you with a couple of things. Another challenge that we are currently working on. So we have all these systems that spontaneously exhibit this turbulent behavior. Well, can we actually control the structure and the turbulent flow and harness it, for instance, to use it, for instance, in microfluidic applications? Experimentalists are starting to make substantial progress with this by using so-called optogenetically modified motor protein. What that means is the following. They take the same protein that I showed you before, but they attach to them light sensitive protein from plants, so that this protein become active only when light shines on them. And then they can use light pattern to actually direct the organization of the microtubules. So here you use a light pattern, microtubule gets organized into an aster by the light pattern, and then it actually follows. You can actually move it around by getting it to follow the light pattern. In this other video, what we have a similar system where only a region, the region in the red square box, is illuminated. So that's the only active region. And so you get the turbulent flows to essentially be only confined to that region and die out very quickly outside of that region where the system is passive. So this is one of the ongoing challenges in the field, is learn how to essentially control and tame these active flows. But, and this is my last slide, you may ask yourself, some of the systems I mentioned are living systems, and so, but I really didn't say much about biology. So what about biology? What is the impact of all this work on biology? Well, I should say that the active matter framework has certainly advanced our understanding of collective cell migration, how cells move together in a number of processes, such as wound healing, cancer progression, or morphogenesis, the process by which a single egg eventually turns into a full organism, as in this example of the fruit fly development. And in particular, in the context of the development of morphogenesis, the question we're really interested in is how is positional and temporal information transmitted and coordinated from molecules or to cells or genes to organs and organisms. And there has been remarkable progress made here thanks to experiments like the ones I'm showing here on the slide. And these are experiments by my colleague at UCSB, Sebastian Streichen, where they're actually able to tag and follow the dynamics of each individual cell. What is shown here is an embryo fruit fly and at one of the stages of the development. And you see these coherent flows of cells that must flow like fluids to get to the right spot. But when they get to the right spot, they have to be able to support mechanical stresses. And the remarkable thing is that they have been able to show that these cell flows are driven by specific protein pattern that control active stresses. and that the kind of fluid dynamics or hydrodynamic model I mentioned earlier can actually quantitatively describe the experimentally measured correlation between the protein pattern and the cellular flows. Of course, there is a lot more work to be done here, but it's a very promising application of Matter's idea to actually understand how you go from a single cell to an entire organism. So with that, I would like to conclude. I've been trying to tell you about this field of active matter and the new physics that arises from cell propulsion with phase transition tuned both by density, but also by new quantities such as motility or persistence of the single particle dynamics. I showed you an example of active particles that organize when passive ones would not, with just repulsive interaction. And I showed an example of spontaneous flow, which is a ubiquitous signature of active fluids. I mentioned how we are now trying to use light and other means to actually harness these active flows and try to produce coherent motion. An ongoing challenge is to try to quantify the relation between information, work, and heat in active system, essentially to develop an effective thermodynamics of this active system. And clearly this is important to understand biological organization and formulating the rules for how to design these machines made of machines, if you like. And then finally, I just very briefly mentioned how active matter is impacting the field of biology, and especially in the context of models of morphogenesis and wound healing. With that, I thank you. And I would like to just show very briefly this. We haven't gotten to take a picture outdoor. Of course, we are no longer just only over Zoom. These are my students and postdoc at UCSB. And I wanted to put up the name of recent collaborators that have contributed to my understanding of the field of active matter in recent years. So thank you, and I hope there'll be questions. Now, am I supposed to stop sharing?
You can decide yourself. Let's thank Cristina Marchetti for her thought-provoking presentation. Now I would like to invite our distinguished panelists who will help us kick off the Q&A section. We have with us today Bob Dorfman, Chris Zarzinski, Ted Kirkpatrick, Dio Marghetti, and myself, but let me say a few words about the process. For this part of the program, I will first invite our distinguished panelists to ask their questions, then I will invite the audience to participate, and you can type your question using the Q&A window that appears at the bottom of your Zoom application. So first, I would like to have, let's say, Bob Dorfman asking the first question.
Cristina, that was an extremely exciting presentation.
Thank you.
And I especially enjoyed the interplay between the microscopic approach and then the mesoscopic approach using hydrodynamics. Of course, my natural tendency is to start with kinetic theory, but I'm not sure that's the right way to start. So let me ask you, when you got interested in the subject, what problems did you consider and how did you approach them?
Oh no, when I first got interested in the subject, we actually did the kinetic theory of rods cross-linked by a very simple model of motors described as sort of a really like a little link that walks on the cross-linking rods. And we actually did the kinetic theory of that to try to derive, we constructed Fokker-Planck equations and then constructed from that coarse grain to construct such equation for density and orientation of filaments and so on. So actually that's how I started. And then right now, I do a bit of both. Sometimes I have students who do numerical simulations. Sometimes I try to start from the kinetic theory and do coarse graining. Sometimes I just start at the macroscopic level, writing down equation phenomenologically. But when I started, I did start with kinetic theory, I have to say.
I probably would also. Thank you.
Thank you.
All right, we have one question for the audience. So let me mix and match. As soon as I receive a question from the audience, I will interrupt the panelists. So right now, the question that I have received from the audience says, could you comment on the current progress and future of searching for equations of states for active matter systems?
Yes, thank you for the question. So let me start telling you about an example. So there has been quite a bit of progress, for instance, on trying to understand the pressure of this collection of self-propelled particles, as I call them, these beads with just self-propulsion and repulsive interactions. There is if you think about it, since these particles are self-propelled, they will exert a pressure just because they swim. In fact, it's called the swim pressure. You know, they transport, I don't want to call it momentum, but propulsive speed across a wall, if you like. And there has been a lot of work on developing a pressure equation of state for this system that has been quite successful. There has been progress on understanding the tension of this phase-separated, the interfacial tension of this phase-separated system, that in a naive calculation based on something like Kirkwood expression would turn out to be negative. And so there was a big puzzle, because a negative tension would not allow the clusters to be stable. But now there's been progress understanding that. Much more limited in terms of equations of states and things like that is the understanding for more complicated systems. I think that's still quite open, as a matter of fact. So a lot of progress for this minimal model of purely repulsive self-propelled particles, not so much for anything more complicated.
This was a question from Jihue Lu, just to give a credit to the person who was the courageous one to start with the first audience question. So, Chris, do you have something to ask our distinguished speaker?
Sure. So first of all, I'd like to say thanks for such a really, really beautiful talk that really highlights kind of the richness of the rich dynamics of active matter. So, you know, as you know, in equilibrium thermodynamics, we have these universal principles like free energy minimization that allow us to predict how the systems are going to evolve, or what states they're going to evolve to. And as you alluded early in your talk, there are no comparable free energies in these non-equilibrium systems. Can you maybe speculate as to whether you think that we will derive at some point, or even are there some kind of governing universal laws for active matter systems? And if there are not, why does everything seem to hinge on this one little principle of detailed balance?
And sorry, and the last thing you said, if there are not?
If you think there are not these universal principles, why is it that this principle of detailed balance is what decides whether you have universal principles or not?
Yeah, so I'm hoping there might be, I certainly hope there will be some more universal principles. And I would say that, so there has been certainly some progress in using idea from stochastic thermodynamics and calculating, for instance, rate of entropy production in order to, for instance, at least try to classify the departure from equilibrium for these kind of systems. And that's, I think, is progress in the right direction, in the sense that, so calculating entropy production by calculating the ratio of the entropy produced in the forward and reverse path, for instance, that at least gives you an idea of whether this is really a not out of equilibrium system or not. And that's one way to look at the systems that I think is quite useful. whether we are going to be able to have something equivalent to a free energy that we can minimize, or something that we can maximize to decide what state of the system we're going to have. I guess I don't think so, because after all, we really only have dynamical equations, and even the steady states here are often, the fact that there is no detailed balance means the steady state can support cyclical currents. On the other hand, you could also say that the very fact of having these self-sustained cycles, that's another signature of the active system. So maybe the presence of a cycle or a surface current is an element that you can use for classifying the system again. So I don't know. I don't think I have a good answer for your question.
It was an open-ended question. Thank you. Thank you for the answer.
Ted, you are the chosen.
Let me follow up on Chris's question. Wouldn't you think, just like an equilibrium system mechanics, when you think about equilibrium critical phenomena, those universality classes are much more restricted or much smaller than dynamical critical phenomena. Every dynamical system has its own universality class. So wouldn't that be true here? It's as you said, it's the dynamics which will control the universality class, and it's going to be different for different active matter systems.
Well, okay, let me say, I think there are some, okay, I'm not sure if I can call them universality classes, but certainly there are classes. For instance, I would say we can classify active matter as scalar, nematic, and polar. And what I mean by that is the model that I described, where we have these spherical colloids that are self-propelled, I would call that scalar active matter. The quantity that I used to describe it, the only order parameter there is really the density, so it's a scalar field, okay? Then I talked briefly about nematic. Nematic active matter, then the order parameter, there is an orientation, but there is no direction, while in the V-check type of model, there is actually a direction, so the order parameter is like a vector, like a ferromagnet. So I have these three classes, and we certainly have some understanding of the difference in behavior between these three classes. But then you are right that if I start trying, so in general, in the few cases where people have tried to say calculate critical exponents or things like that, those things are not universal. The critical exponents are not universal. Another example is we have actually done some work on understanding this turbulence as effectively, in 2 dimensions at least, as a KTP type transition, a defect unbinding transition driven by activity. And there is a lot of analogy with the BKT transition. And there is also a jump in stiffness, but the jump is not universal as it is in. So there are a lot of analogies, but often the behavior is not universal. So I think you're right, maybe because it's all driven by dynamics.
So the next question will come from the audience, and the audience is our distinguished colleague, Dave Livermore. So the question is, Cristina, do you see relationships between these and other models that capture emergent behavior? For example, models of traffic flow, either on a road or on a road grid, and exhibit a phase transition between free flow and traffic jams with repulsive interaction.
Oh, yes. I mean, the models, what I call the motility-induced phase separation is essentially a traffic jam. It's a traffic jam, not on a grid, but it's a traffic jam in a continuum level, but it can be described in some senses very closely related to models of traffic jams. Also, for instance, models like the ones I don't think I can say I described, I kind of alluded to, have been used to describe, for instance, human crowds, how human crowds can get stuck by an opening, or the behavior of human crowds in what is called the mosh pit, when they go around essentially in circle that establish spontaneously, just like bacteria spontaneously exhibit vertical type behavior. So that's exactly kind of one of the ideas of the field, that there are certain type of models that seem to be able to describe collective motion on many different scales and in many different systems. Now, the challenge comes when, for instance, if you write these continuum type equations, the way that again I've mostly alluded to, I would say, the challenge comes when you want to try to relate the parameters of the equation, such as, I don't know, the viscosity in the Navier-Stokes equation or the stiffness of the pneumatic, to experimental quantities. Because that is much harder, and generally, there are a few cases in which we know how to do that, but generally we don't really know how to do very well. Not sure if I answered your question. If not, feel free to complain in the chat.
Yes, Dave, you can come up with another question if this is not clear enough. And I would like to invite Dio to see if he has any question.
Yes, so thank you for this talk. So in your talk, you nicely navigated between discrete models, which are inherently the particle systems, and continuum models. And the question I have is a bit philosophical, but I want to see it in the context of active matter, is the relation between the two types of models. Specifically, whether there is enough progress in your mind in relating parameters of continuum models with the discrete models. And I will refer here specifically to your first example of new physics, spontaneous assembly without attractive forces.
Yeah.
So one can write down, of course, so you wrote down a discrete equation for the velocity of each particle, which of course one can naively perhaps take in the continuum limit. So the question is that, let's say, reasonable, and has this approach been successful? That's my first question.
No, I mean, in the context of that system and that example, I think, yes, the approach has been fairly successful, meaning we can, as I was taught to do when I was a student and then a postdoc at IPST, we can actually write down a particle-like equation, and then we can actually go from them to sort of a kinetic theory, and from a kinetic theory to sort of continuum hydrodynamics. We can do that for these self-propelled particles. Now, usually you have to make approximations, and for instance, in this case, maybe low density or weak interactions, and so the parameters that you calculate may not be quantitative, precisely quantitative fit the data. If you compare with simulation, though, the equation that you obtain really do a very good job at predicting the behavior of the system. You may have to adjust some of the parameters because you can only calculate them at low density, but in that sense, you really know pretty well how to do this. Now, if you, at a very different level, say you want to take these continuum equations and use them to describe cell migration in wound healing, okay? Which is actually very interesting. What that mean? It means that when a cell monolayer expands into free space, and how do they communicate to know where they go and how to move coherently? So there, actually, the idea of and actually you can do that. You can actually do a pretty good job at describing the collective behavior, but indeed relating the parameters of the continuum model to cellular properties, that's very much an open question. That's something we really don't know how to do yet. So it depends how complicated the system is, I would say. For some minimal models, we know how to do this. We can do it as well. We can do it at least as well as you can in equilibrium. Okay. But for more complicated systems like a biological tissue, no, we're still far from being able to do that.
Allow me to ask a second question, can I? Just
course,
as
please,
a continuation,
please.
just
Yes.
So The second question I have is more technical, a little more specific. So again, in your first example, you state at the end that, of course, one can use non-spherical particles or more complex interactions and so on. And this has been done to some extent, I suppose. So my question is this. So suppose we don't use identical particles. So that means that we allow for some diversity. Let's use that word. Is there a simple message how diversity affects assembly?
Mostly, it will mostly affect the property. So, okay, let me give you a simple example. If you, let's say that you have your spherical particle, but they're polydispersed, okay? The only thing that will change is that if they're monodispersed, the clusters will be pretty much crystalline, perhaps with grain boundaries, but pretty much crystalline. If they are polydispersed, they'll be more disordered, more glassy-like. Okay. If you mix, say, self-propelled rod and self-propelled discs or something like that, then shape can actually also drive phase separation. So you might have regions of discs and region of rods or something like that. So what you call diversity, if it's diversity of shape or diversity of motility, actually.
Of motility, yes. Mm-hmm.
Diversity of motility can also drive phase separation. If you put in a box particles with different motility, then I can remember, I think the faster one will aggregate at the boundary and the slower one will remain inside. Variations in persistence will have a similar effect, in persistence of the dynamics. So yes, and actually that's very important if you want to start thinking about, again, simulating, modeling systems like collection of living cells. They are certainly not all the same, not all the same size, not all the same shape, not all. So, but the other thing that it's really also very important and still relatively unexplored is actually noise. A lot of these continuum models, especially, have been wrongly studied at the deterministic level, meaning people study PDE with no noise in them. And clearly these systems are very noisy. And in this nonlinear continuum PDEs, the noise can actually make a difference very often. It can actually sometimes select states. It can sometimes drive order or disorder depending on the situation. So that's something.
But you mean noise as a forcing in the equation,
yes,
actually,
yes.
Not random initial data, not in the initial data of the equation.
Random initial data, of course, is there, and everybody thinks, okay, instead of noise, I just put random initial condition. But the noise generally, if you derive the equations a la Bob Dorfman from a microscopic model or Alted-Kirkpatrick, you can also try to derive the noise, and then the noise will generally be multiplicative, actually. And so that kind of noise can really make a difference. And that's relatively unexplored, actually.
Thank you.
I have a question, but before I ask my question, I would like to brag a little bit as director of the Institute for Physical Science and Technology. I would like to mention to you that our institute is the house of the first hub of quantum thermodynamics in the US, thanks to the leadership of Chris Dzerzhinsky and Nicole Junkel. And I wanted to mention to you also our efforts in the Institute on aspects related to quantum information science. Many of us are working nowadays on quantum algorithms. And we were discussing earlier, and you mentioned the connection between statistical physics and machine learning. And I wanted to ask you, do you have any insight about potential influence of statistical physics in the design of quantum algorithms? I think this is something that it will be very relevant, maybe, given the fact that optimization is so important on the design of quantum algorithms. Is my intuition correct?
I don't know if I can really answer this question, but let me just say that I have several excellent colleagues here in Santa Barbara working actually on atomic and molecular physics. And also people working on quantum circuits from a theory point of view. And on these ideas, how if you have a quantum system that, it's in non-equilibrium, it will spontaneously become entangled and therefore essentially thermalized from their point of view. And therefore thermalized, which means from the point of view of people working in this field, it's uninteresting because it's no longer quantum. Okay. And one way to keeping it from thermalizing is a sort of repeated random measurements. This is work that was done by Matthew Fisher. And we've been discussing a lot to explore the possibility whether there could be some correct connection between the repeated random measurement here that keep the system from thermalizing and the persistence of the dynamics in the active matter systems that is something that has the opposite effect and drives the system out of equilibrium, because the persistence of the dynamics is, it's not, of course, quantum measurement is different because it disrupts the system. But anyway, we are exploring connections. There's the possibility of connection between this kind of phenomena. So I don't know, but that's
yes, but it is possible at least. It's
possible.
We have one more questions from our colleague.
Let me just say one more thing. Another possible connection that might actually be a real one between this kind of systems and systems that have been studied more by people working with quantum phenomena has to do with a sort of there are when you have a mixture of two type of active systems that have what we call non-reciprocal interaction, meaning A does to B and B does something different to A. And the typical example in statistical physics can be called the prey-predator system. But you can, especially in these phoretic active colloids, because the interaction are mediated by a non-equilibrium medium, there are actually effective interactions that don't obey Newton's third law. And then you can actually design a system where you have the two species, and one will tend to run away from the other, and the other tries to catch up. Okay. So these non-Hermitian interactions and their description has close connection with non-Hermitian quantum mechanics, as a matter of fact. And there are some real connections one can actually make. Wonderful. You get traveling states that are related to the so-called time crystals that people in quantum mechanics study and so on. So that's yeah.
We have one more question from our colleague Dave Livermore. So he returned with another question, and the question says, does the power spectrum of the turbulence in quotation that you showed exhibit power law?
So one thing that has been shown is that the distribution of vortex size, flow vortices, of the size of flow vortices is exponential. Not a power law, but exponential. So you don't have vortices of all sizes with a characteristic length scale, which is this active length that I mentioned. This has been shown in simulation and seems to also be the case in systems which are quite diverse, these microtubule kinesin suspensions, as well as the, for instance, monolayer of cells. If you're asking about the spectrum, say, of a kinetic energy, yes, there is a power law, which is different from Kolmogorov power law. I showed you a number. I don't actually remember the number I showed right now because it's not the same in every system. And so, and so possibly is not universal. I mean, probably, not possibly, probably.
Could I ask a question?
Yeah. Of course, please.
In your motility phase diagram, you mentioned the glass phase. Could you say some more words about that and what its biological realization is?
Yeah, the glass phase, well, I think the glass phase is quite boring because I'll tell you why. I mean, I say that.
Jam particles?
The glass phase is a phase of giant particles. So your question has two parts. The first part, the glass phase, I think the glass phase itself of these colloids is relatively boring. And the reason I say the glass phase is boring is because the glass phase can actually be understood in terms of an effective temperature, as opposed to the phase separation that cannot. You can actually understand the glass phase in terms of an effective temperature. And so it's like particles that are jammed, but they jiggle around a bit because there's this effective temperature, and they do exhibit what people in glassy system call, oh God, I can't think of the name, these collective rearrangements, what are they called?
Are there astronomically large relaxation times?
Oh, I don't know. There are large relaxation times. I don't know that. I mean, I don't know that anybody has quantified things like, but yeah, there is a lot of behavioral glassy system. There is aging. There are large relaxation times. Its behavior is very similar to that of other glasses. That's why I say it's boring. Okay.
Mhm.
Now, in the biological context, is quite interesting because in the biological context, there has been actually quite a bit of work on glassy states, or actually liquid glass transition of cell layers, of living cells. And what is interesting in that case, these are liquid glass transition of so-called confluent cell monolayers, meaning a confluent cell monolayers means a layer of cells which are completely packed. There are no gaps between cells, so the packing fraction will be 1. So there are no variations in packing fraction. So the transition is not driven by, say, packing fraction, but it's driven by other processes that control the tension of the cell edges, essentially, and is quantified, and you can actually quantify it in terms of cell shape. Meaning, if you measure the mean cell shape, the glassy state will be one where the cells are fairly, let's call them fairly compact and round, and the liquid is one, and in that case, the cells don't exchange neighbors, then they are solid-like and so on. And the fluid state is one where cells are fairly elongated and they exchange neighbor frequently. So cell shape is a metric for fluidity, if you like, which I think is a very interesting point. And it's a glass transition driven really by a new quantity. Of course, motility also can tune the glass transition in these systems as well.
Okay.
Are there any other questions, either from the panelists or from the audience? Let's give some time. Bob, do you have anything more to add?
There were so many questions that occurred to me in the course of Christina's talk. But the one question that struck me was also asked by Chris, namely some universal strange kind of thermodynamics properties of these kind of systems. Whether they will exist, I don't know, but it's certainly tempting to ask if there's an entropy functional or a large deviation functional, something that might put these different phenomena in a more general context. Is that possible, do you think?
I think, you know, I don't really work on that, but there are people who are making progress in this direction by looking at entropy production, as I was saying, and calculating and measuring even actually experimentally the entropy production for this active system, and finding some you know, I'm not going to say some not universal features, but certainly finding very commonality for now. Perhaps we can call them or something like that. Okay, so yes, I think we are heading in that direction, and I think hopefully we'll get there.
Thank you.
Now, whether this will actually also really apply to things like living cells, I don't know. I will.
Let me ask this question from the audience because it is connected to a question that I had during your talk. The question says, is there any relevance to the bacteria quorum grouping that protects them from external chemical treatments?
What does he mean by relevance? I'm not.
For instance, when you were talking, I don't know if he is referring to the same thing. When you were talking in your talk, you mentioned about E. coli and you saw some experiments.
If this refers to the fact that there is this the bacteria also communicate chemically, for instance, and as a result modify their motility and tend to aggregate. Yes, it's very similar. As a matter of fact, there is another way of modifying the motility that can lead to aggregation is through sort of chemical interactions, as a matter of fact, or quorum sensing, as is called in the context of bacteria. It can also be described by a theoretical formulation very, very similar to the one we use to describe this motility-induced phase separation. And in fact, this has actually been done, for instance, by Mike Cates and his collaborators.
Mm-hmm.
So yes, it's a very similar.
Well, what an enthralling session that was. I would like to once again thank our distinguished guest and lecturer, Cristina Marchetti, the generosity of the Pai family for making this lecture series possible, and the panelists and general public for making this a memorable occasion. So,
well, thank you very much for asking me, and it was also great to see some old friends or new friends, recent friends.
And I promise you,
some that I have not seen in a long time.
You will be invited back in person because we love your lecture. It was exciting to me and to our colleagues.
Thank you.
And so we would love to have you on campus next time.
Thank you. That would be great. I actually would love to visit the University of Maryland. Can I ask one quick question?
Of course.
Ted, where are you now?
Right now, I'm sitting in the mountains of North Carolina.
Okay, I see. Is that where you are living, or? Yes, I guess.
You know what? We have a house in the mountains of North Carolina, and we also have a beach house in South Carolina.
Okay, all right. Maybe I should visit. Maybe I should visit that too.
Helen is in South Carolina.
Okay. Well, say hi to her too for me. Okay. All right. Well, thanks again then.
This concludes the 28th Dr. Shih-I Pai lecture. Thank you very much, Cristina. That was exciting.
Thanks, Cristina.
Thank you very much.